Wonder if Stu's joining us today, Stu and Catherine. I don't think so, but I haven't talked to them.
He accepted the invite, so
Oh, well, no, maybe he will then.
yeah,
And this is
great,
a topic you should find pretty interesting. So it'd be nice if Stu joins us.
great. Okay, I'm going to just kick it off then. I'm Charlie Munford. This is Agential Topics. This is a theoretical biology discussion group centered around Talking Octopus's mission at this point, which is to create a physical model of agential biology. This group was actually started by Wayne Lewis, who is here today. And anyway, we today have a special guest, Daniel Phillips and Denis Noble, have both joining us and have some incredible, an incredible paper, or series of a paper and then a letter, and in response to the letter that we're focusing on today, which is all about extracellular vesicles and the pathway of inheritance that goes from the soma to the germline. So, Daniel, if you want to say any words to begin, take it away.
Okay, sure. So I should probably introduce myself, right? So there's lots of new faces and people I haven't spoken to before, haven't met before. Some of you I recognized. Hey, Wayne, good to see you again. So I'm Daniel Phillips. I'm actually a PhD student here in Oxford, where I do some of my work with Denis. I'm not in Denis's group. I actually just happened to meet Denis on the street and just got talking to him by sheer luck of chance. And the first thing we started discussing actually was this new schema that seems to be emerging in physiology, a new cellular communication platform based on these tiny bubbles called extracellular vesicles. So joint interest is obviously physiology, but evolution, and you could call that, I suppose, the physiology of evolution. So the context of a lot of things we think about, all of us in this group, are how does things which happen in the body relate to the grand scheme and the grand trajectory of evolutionary processes. So we were learning a lot about how these tiny bubbles, extracellular vesicles, do work within the body to send signals from one tissue to another, from one cell type to another, to adapt to changing conditions of the environment, be it diet, stress, temperature, learning, just any myriad of things. And the question then was, does this have any role to play whatsoever across generations? So the reason why I started looking at this actually was maybe long-winded. I won't go too much into it, but it was actually in the context of the brain and learning. So I was really, really interested in how does learning happen? How does memory work? And when I was an undergrad, I was committed and sure that I would be working on animal behavior and learning probably for the rest of my career. And I learned that how a lot of learning happens at the molecular and cellular level is with tiny bubbles communicating across synapses, sending messages, sending almost a memory of what's just happened in one neuron to the other to create this system of shared knowledge, you might say. But then I realized that these bubbles can, if you like, lose track of where they're supposed to go. They can join the highway of the body, join the circulatory system, and they can send messages all across the body. There is really no limits. And then I learned actually one part of the body that uses this communication system the most is the reproductive system. And these bubbles can come from anywhere in the body, firing left, right, and center. But the most interesting part to me is when they come from the body, go all the way to the reproductive system, and enter the sex cells, the gametes, the sperm, or the egg. And the reason that was so interesting is obviously from a historical perspective, evolutionary theory, there shouldn't be any communication with the body and the sex cells. The sex cells from the very earliest stages of development should be splitting away and doing their own thing, and they should be totally isolated over here. So the fact that there can be bubbles, physical bubbles, not just some information concept, but real physical things, making it all the way into, particularly the sperm is where lots of the research is being done, was just on the very face of it quite mind-blowing. But the fact that they were probably above everything else, these bubbles containing nucleotides, genetic information, just made the story so much more juicy and fiery. And then to top it all off, it wasn't just DNA, it was also RNA, non-coding RNA, which is where my interests really lie, because non-coding RNAs are extremely potent. They can do almost everything you would want for an effector molecule to do in the body. They can be genetic information, they can remodel genetic information, they can reshape and rebundle and just everything you would want to do. So this is how the story now stands. If you've all read the paper that I think was circulated, whenever something happens in the body, there is a basis of the physiology using extracellular vesicles to communicate how the physiological system should adapt to that change. And that same message gets sent to the germline, and it kind of reprograms the germline to recapitulate such adaptations in itself. And some of them can be inherited, or at least predispose the next generation to at least partially phenocopy some of those changes. So I think the big question is now, does this matter? In what ways does it matter? Where does it matter? To evolution only? To clinical health? And it just opens up a real Pandora's box for concepts in evolutionary biology, concepts in health biology, so many questions. So I think there's real deep raw materials for a good discussion here.
Maybe biosemiotics as well, actually. So you have communication between organisms. Something I've actually even talked about is that there's a big difference between having a Zoom meeting with someone and having a face-to-face, or having, so when you're actually in the room with someone, in addition to, body language and things like that, you're constantly exchanging these vesicles with the RNA and DNA, and that have all kinds of signaling, communications significance. I don't know if you've looked at that at all. But.
Yeah, I think I became really interested in biosemiotics actually even before I was an undergrad, and you're spot on. This is all membrane surface level things. So everything that happens with these EVs is one membrane recognizing another membrane. So there has to be some kind of meaning generation or meaning transference happening there. I think you can have discussions, and you can certainly do research on extracellular vesicles without thinking of it in this way. But at least for a hypothesis generating theory, sometimes I'll try on the biosemiotics kind of hat.
Just for many of us in this group, it's really all about cognition, and the biosemiotics is fundamental to cognition. That's why I kind of brought it up.
I have a question about the Weismann barrier. So this is obviously like hugely significant because it violates the Weismann barrier, which was, I guess, for almost 100 years considered to be this barrier between the information transfer from the soma to the germline. But and this may be a question for Dennis. I mean, it's kind of a historical question. So I looked it up, and among eukaryotes, we have protists and plants and fungi, and none of them respect the Weismann barrier. And then among animals, we have cnidarians and flatworms, and they don't respect the Weismann barrier either. So I just wonder how was it that the Weismann barrier became so important? It became this like bulwark of evolutionary theory. If it doesn't apply in most eukaryotes, let alone prokaryotes, which do horizontal gene transfer, how did it come to be so like conceptually entrenched?
Can I comment before? I'll raise my hand.
Yes, if that's directed to me, I think I can give a reasonably straightforward historical account of that phenomenon. And it begins with 1866, So about 7 years after the publication of The Origin of Species, 8 years after the joint presentation to the Linnaean Society by Charles Darwin and Alfred Russel Wallace, outlining the idea of natural selection, which they came to independently. Darwin, of course, because of his work in the Galapagos, and Wallace because of his work over in the Malay Archipelago, or however you pronounce that. Now, that meant there was, in 1858, complete agreement between Darwin and Wallace on the idea of natural selection. Now, in 1866, Wallace started to wobble on that issue. He actually came to the conclusion that humans were not subjected to natural selection, which is a very strange position. It's almost Descartes, going back to 1665 or so, with his treatise on the fetus, that in some way or another, how best to put this, that humans were special. I mean, there he was actually asking the question, how on earth does our spirit, somewhere here, I don't know where, have an effect on our nervous system? And he never really properly answered that question. But he came to the conclusion that animals didn't have it at all, because they, after all, were not going to go to heaven. But that's a religious background. Now, to return to Darwin and Wallace, 1866, Darwin was angered, very strongly so, and you'll find that in the biography by, can't remember their names now, but it's a very substantial biography of Darwin and regarded as the classic. Now, Darwin had already started thinking, could it be that natural selection alone explained everything? And he came to the conclusion that there were some phenomena that could not be explained by natural selection alone. Those included his own conviction that use and disuse was somehow communicated to the next generation. He was, in that sense, a Lamarckian, although he never used that word. Indeed, nobody in the 19th century would have done, because it was an idea that was general, not specific to Lamarck. Now, Darwin then set about dealing with Wallace, because he felt that it was a great tragedy that the agreement between them in 1858 with their joint presentation to the Linnaean Society had led to a split that Darwin was convinced was incorrect. Now, Wallace himself was wobbling around. You'll find that in the comments on Wallace's ideas in George Romanes's book or trilogy, Darwin and After Darwin, published after Darwin's death. But the key comes then on what happens, the consequence of Darwin's reaction to Wallace in 1866. He started writing, and he produced 2 or 3 really further books. The Domestication of Animals and Plants, in which he outlined his ideas on how could it be that some communication could exist between the body and the germline. And that's where he formulated his idea of gemmules. Well, I mean, what Daniel and I say in the paper is they are almost functionally doing what Darwin postulated as gemmules. And he always admitted it was only a postulate. He imagined they were far too tiny, and he was quite right, of course, if they are the vesicles, that they're far too tiny to be seen with the light microscope. But that's the background. And when Weismann came along, the rift was even stronger. Weismann came along with his idea just after Darwin's death in 1883. 1882 was when Darwin died. And that's when he proposed the barrier idea in order precisely to exclude Lamarckism. So the logic in their minds was that Lamarck is wrong. To the extent that Darwin has followed Lamarck, he must also be wrong. And that's what generated this split. And Wallace, in effect, became the first neo-Darwinist, supported then by Weismann and by Galton, while Darwin, sort of on his own, until he got George Romanes to join him, ploughed his own furrow, disagreeing them very strongly. The second set of books was a 2-volume publication in 1871, so 5 years after, no, 3 years after the domestication of animals and plants, in which he outlined a general idea. And what he did there was to explain his idea of sexual selection. So what was happening was that Darwin was postulating a more extensive set of processes that interact with each other in evolution, while Wallace was hardening on the idea it can all be explained by natural selection. That's the origin, historically, of the split.
I can't find the raise my hand button right now. One thing I wanted to comment about just real quickly is that, I think a lot of times it's certain ontological and metaphysical commitments that kind of force you to lock things into certain frames that would, they're trying to simplify it. They want some kind of a simple mechanical, essentially Newtonian paradigm kind of explanation. You see the same thing in physics, where, if you aren't willing to accept that possibility has ontological status, and some Stu would talk about as well, then you have a real problem between the Copenhagen interpretation, where you have this nice linear evolution of the wave function, and you have this really nonlinear process of wave function collapse. But once you're willing to accept that possibility is something that's actually part of reality, this goes away. And then, you have these same kind of problems in biology, where, you're committed to this kind of linear picture. And within a linear picture, if the germline is segregated, now you can have this nice, simple, easy to understand linear process. And this is everything, and there's nothing else.
Mary?
Hi, everyone. I didn't think I was going to be asking my putting forth my wild conjecture so early in this process, but here goes. Charlie, you can help me with this. So I was raised by a crazy Italian engineer who taught me that when you walk into a room and you see a car taken apart, you just have to start thinking about what all those pieces do. And the car being taken apart was all too frequent in my house with my 4 brothers. So I look at the cell and I see a lot of equipment. I see a lot of optic equipment, and I see a lot of things that could be used for optic processes. And I'm wondering, and so I've been talking about this with Marco and Charlie and Wayne a little bit and Rafe a bit. And I've been meaning to reach out to you, Bill, because this is related to the epigenome. Is that how you say it?
Genome?
Anyway.
The cell.
Yeah. Okay. So anyway, I've been working with this idea that memory is a complex process, but it involves somehow recording, creating some level of recordings on the membrane, and the vesicles being involved in this to some degree. And this goes all the way back to ideas that I learned from Albrecht Buehler at Woods Hole, and I don't know, in 1986 or something. I'm wondering, when I look at the cell, I see that the cell is in many cells, if not all cells, are at least one lens. Cells are able to cohere light and to polarize light. And they have these wonderful membranes that are multi-layered, that have the embedded membrane proteins, and they have the polysaccharides, and there's structured water. And I'm wondering if and then there's also, well, the structured water is a big part of this, but I'm imagining light energy coming into a cell and moving through the cell and being changed in some way. And then there's also the spiralization of polarized light using the chiral molecules. And the membranes themselves are also metamaterials. They can change their electromagnetic properties at will. And I look at all of this and I see the opportunity for light to come in and to be captured, focalized using the lens capacities of the membranes, and focalized on membranes, and then the membranes with their layers of the proteins, the structured water, the polysaccharides, being in some way let's just call it a recording plate. And this is where we have a little pun of the cell being a camera. Cell and camera meaning the same thing, right, Marco? Cell and camera both mean room. And the membranes literally being like meme brains. I'm trained in somatics, somatic awareness, and we were firmly taught when I studied at the same time as I was studying cell biology that what you learn at the cell level, the cell wants you to understand at a higher level of holon of the organism. So perhaps at some level, the cells helped us to learn those words so that we would get the pun of the meme brains. And that you can even look at the Hemroff, Chalmers, Penrose ideas about the microtubules and see the microtubules as having a capacity in some way of transmitting holographic images. So for folks who don't know, holographs are created when polarized, coherent, phased light, laser light, is actually, there's an interference paradigm created, and then there's a recording on a translucent polymer. And so I'm looking at all this and saying, well, using the way I was taught to look at the cell, I see all of this equipment lying around. What is it actually doing? And I wonder if anyone in here has any ideas related to this. Oh, and also I'm really interested in the membrane rafts, because membrane rafts have somewhat of a kind of, I wouldn't say a permanent quality, but they have a enduring quality. So this is my wild conjecture, and the more I look for stuff, the bigger my research bibliographies are getting, indicating that there might be something here. And I know that everyone in here that I know personally, I don't know Joshua or Omar yet, and Daniel, it's great to meet you. I'm sure everyone has something to say about this. And now I'll shut up.
All means. Can I try and tie those things together with a question, which is, so RNAs and DNA, and in the letter from Sun and Liu and your responses to it, you talked about free floating DNA in the bloodstream and how that can be instructive to the germline as well. So RNA and DNA, I think, obviously the dominant conception of sort of their ontology, what they are, is that they're involved in self-replication. And Dennis, you've very cogently challenged that in a lot of public ways. But I think I think the alternative is that they're memories. I mean, if they're generated by the body to sort of interpret the world, and then they're communicated as interpretations that then further instruct, we have a name for that, and it's memory. And it seems to be not at all implausible that if memories can be contained in the configuration of neurons, they can be contained in the configuration of other things, just in principle.
Well, in this idea that I'm constructing, RNA could be also involved various nucleic acids could also be involved in this memory kind of complex or cascade, if you will. Obviously, if we look at Luc Montagnier et al.'s research and the ideas of the structured water folks, I call them the Italians, and Mei-Wan Ho, and also the work that Gerald Pollack is doing, there's a possibility that structured water is holding a structure or creating a resonance that allows nucleic acids to be attracted and to act as a template for creation of, I believe he was doing it with DNA, but you could also see it as RNA, and there could be some co-templating of of the structures that happen if there's a cohered holographic image that's captured on a membrane, for example. And the RNA could either be, the water could be the negative for an RNA, the structured water could create a negative for an RNA molecule that could then be sent via vesicle. Oh, I also forgot to say that the way the idea of the light transmission down the microtubules could work is that the let's call them the memory rafts, for lack of a better term, could be used to actually, if light goes, if they're used to focalize or to retransmit an image, then the microtubules could be used for sending that image. And I'm just trying to think about this with as little interference with the whole, quantum effects as possible, to see how far we can go using what we know about physics and electromagnetism without going into that realm.
To answer
Ray?
Charlie's question,
Oh,
So,
I was going to say, to answer Charlie's question about genes and memory, RNA and DNA and memory. Genes, all genes are tools of the cell. They're retrievable and deployable memory. And for RNA and DNA, the memory that they encode are prior solutions to environmental problems. They're a record of the problem-solving trajectory of the organism over time and the experiences that it has had. And that is exactly why in cancer and also in normal cells, cells will reach back into their toolbox, into their genes, and they will redeploy genes that have been used before. So in epigenetics, we have many circumstances where genes are silenced, repressed, or then de-repressed. And many times what you have is a skip between of a solution that befitted great grandparents being redeployed under contemporaneous stress. And so, yes, they're tools of memory. And so is every other process within a cell. The cell is a whole. It functions as a whole for reasons that we have yet to understand. And within that cognitive base, everything is a tool of the cell. And all stimuli are being utilized and assimilated continuously from the environment into the cellular environment.
thanks. Rafe?
Yeah, your question about, let's say, maybe the dual nature, not either or perhaps, of genetic material, self-replicating genetic material, and whether or not that is a functional memory in an organism. I happened to put in Daniel's papers that you shared, Charlie, into ChatGPT yesterday to get an understanding of them. And I went down a rabbit hole and asked it, is memory dependent on prions or self-replicating structures? Because it had kind of seemed to indicate that was an evidence. And the conclusion that ChatGPT reached is, yeah, both mammalian memory and neuroplasticity, as well as cellular memory, is partially dependent on like prion-like proteins, if not prions themselves. And so to me, that's just validation of maybe the dualist interpretation, or the dual interpretation of these structures as, yes, they're self-replicators, but they're also functionally memory, both at the cellular and the supracellular level.
Why did you think it had to be prions, Rafe?
I'm just fascinated by prions. And I don't know why. I noticed that kind of popped out at me as so I don't know why I'm fascinated by them. And I wanted to find evidence. I wanted to see if there's evidence that they're more than just disease generating structures. Like, and so that's kind of the inquiry there.
Wayne?
Yeah, just a very quick comment. One very general thing in all biology and in cognitive science is acceptation. And what you're going to find out, of course, we always have to do all the fundamental work, detail these various processes. But what you're going to find, I believe, is that anything that's exposed to life and that can be used in a way that's adaptive or problem solving is going to be used. You're going to find down the road, I mean, the DNA can act as a molecular wire, and there's all different kinds of things that all these sort of modules of cells and organisms can do. And anything that can be used by biology will be at some point. So it never surprises me. None of this stuff ever surprises me. These are just, life is just constantly finding new capacities for existing structures.
Well, it strikes me as a little bit of a paradox that, the animals, like this one tiny branch of the tree of life is animals that have something like a Weismann barrier, and they seem to be the most complex and also to have the most general intelligence. And so it's a little bit of a paradox to me, because I think of intelligence as stemming from this other cognitive source, that we have so much protection for the like fidelity of inheritance compared to plants and fungi, stuff like that.
I think you're going to end up finding that the degree to which the Weismann barrier is really respected in these organisms is overblown, because what you look for is what you're going to find. So there's a large pre-commitment to this sort of Weismann barrier. So we really don't look for things, in large research programs that would disrespect that. And what we're going to find increasingly as we begin to look is that we're going to find more and more examples when our eyes are actually open to being able to observe them. And we actually look.
Right, but still, like, just imagining the opposite. Like, why is it that the most complex animals don't just profligately incorporate DNA, from everywhere? I mean, we could be set up a different way, right? We could be set up without a germline at all, and like just our fingernails could bud into, new Charlies, and we could take up more DNA. Like plants often have huge genomes, and planaria have really messy genomes.
Yeah, this is the question I asked Mike Letter. I mean, the way the planaria work seems so friggin' robust. It's amazing to me that all organisms don't work that way. Yeah, that's a tough question.
I mean, like, if we're cognitively, and we have more, we can do more cognitively, right? Why is it that if genes are tools of cognition, why is it that we don't just have way more genes? Like, it's back to the question.
Well, it may be the case that we just have a bunch of other different things that we can do, and we've sort of, to use the word higher organisms, we've sort of canalized certain types of memory processes to DNA, and there's a bunch of other florid things that we haven't even really detailed yet, that are involved. I mean, in Jablonski even, and et al. Detail some of them in that evolution in 4 dimensions, but there's probably many that we're not aware of. So maybe it's a cognitive advantage to sort of maybe close off the germline to a larger extent than it would be in the lower organisms, because you have all kinds of epigenetic inheritance mechanisms available to you, such as, you know, social and behavioral things that you don't have available maybe in the lower animals. So that may be the reason, but it's completely speculative. Of course, you have to do a lot of research to answer that.
Yeah, if Henry Heng were here, I posit that he would argue that it's really important for higher organisms with sexual selection. Sexual selection is meant to stabilize the genome and the germline. And if we had all these kind of free floating mechanisms and memories that overloaded the genetic mechanisms, it would be literally genome chaos, and that would not lead to an enduring species. So that's a different way of interpreting it.
It makes logical sense to me that since membranes are obviously to some degree transmitted during embryogenesis, well, during reproduction, and the vesicles, the whole idea of the vesicles happening, being transmitted, you know, at all of these different levels indicates to me that there are multiple ways. And Bill is the first person who talked to me about this and got me thinking about this. There's that membrane is everywhere. Membrane's happening everywhere. And Charlie, you say this too, don't you? There's multiple ways that the thing that we call memory can be transmitted from a parent to an offspring. And I mean, has anybody done this? I mean, maybe Neil deGrasse Tyson or somebody has worked up how many miles of membrane we have inside of our cell, how many moons of Jupiter we could cover with all of the membranes in a single cell. And when you think about the ubiquity of this substance and the fact that the glial cells are considered to be so important in memory, and you know, they're basically just lamellar, like memory floppy disks, the way I look at them, possibly. I think the vesicles are probably a very, very good target, a good suspect for, and the membranes themselves for being integral and central to memory.
Dennis?
Yes, I'd like to come in on this question of where memory is. I did a calculation a few years ago comparing the 3 billion length genome with the number of bits of information you would need to represent the rest of the cell, and particularly its membranous structures, which in eukaryotes is vast. The cell is packed with membranous processes where all kinds of things are going on that depend on those membranes, and for which, of course, there are no genes. There's no genes for phospholipids. And it's very easy to show that at a resolution, it's obviously resolution dependent. Any image transmitted digitally is going to depend on the number of pixels you're going to allow it to have. But by any criterion of over what sort of distance, tiny distance, is information still relevant, you can easily show that there's more information passed on in the mother's egg cell and its membranous structures than there is in the 3 billion base pairs of DNA. It's just that it is not easy to represent simply digitally because, as I said, it all depends on the resolution at which you represent it all. But there's no difficulty in saying that the memory of the cell is as much in the rest of the cell as in the genome, and possibly even much greater than that.
That's exactly how I think about it, Dennis. Thank you for putting it so succinctly. Now, if you could just tell me how many moons of Jupiter the membranes in a single cell would cover, I would know. Just kidding. The surface area when spread out of the membranes in a cell would be an interesting number to know for a typical eukaryotic, say, any particular cell, like a human skin cell.
Well, if you're going to do it at a molecular level, it'll be trillions, or at least.
Right.
As I said, it all depends on the resolution. And when you get below a certain resolution, of course, it's an explosion of total amount of conceivable memory.
Yeah.
And that's why it's so difficult to answer the question,
There-
how much does it actually correspond to? It just happens that the genome is easy. You can calculate 3 billion, it's 3 billion bits. As simple as that. Or
There-
however it works out. I can't remember how you do the conversion to binary now. But the fact is that the rest of the cell has, it seems to me, at least as much as, and probably at the right molecular level of resolution, much more than the genome. Now, a lot of that information may, of course, be somewhat irrelevant. It may be just you need rather a large number of molecules to carry out the processes, and therefore, does it matter that you've got so many of those and very few of some other? This is a minefield of difficulty in trying to calculate. I think all we can safely say is there's at least as much transmitted in the rest of the mother's egg cell as in her DNA.
That's perfect. Thank you, Dennis.
I'd like to ask a question about that point, which is just, it seems like in processing information, certainly in computers, there's all kinds of ways where tremendous amounts of information can be compressed to take up much less space, right? And we're finding similar things happening in the brain, right? We find these processes where there's certain kinds of compression going on. And so my curiosity about this has a lot to do with the question of whether there's something more, perhaps, I was just playing devil's advocate here, just if there's something more perhaps to the information contained within the DNA in some sort of coded form or some kind of compressed form that we're not onto yet. So just playing devil's advocate, Dennis, on your very good point.
So, Joshua, are you familiar with the conceptualization of the DNA as being electromagnetic antennas, transmitters, and receivers?
I've heard of it, but I haven't read into it.
Have you read the absolutely wild and out of control Montagnier paper about replicating DNA using harmonic resonance from one vial of water to one that was sitting next to it?
No, but I'd love it if you sent a
Oh
link
my God, I'll send it to you.
Okay. But I'm curious about Dennis's response and whether he was thinking about that kind of thing when he was doing his calculation about bits and the amount of information that DNA can contain.
Well, I think it's interesting that because I'm thinking of the transmission of memory or information, which I think of as like Bateson information, like influence from one level to the next. So this paper is about the soma to the germline, which is the phenotype to the genotype, basically. Although we're talking about epigenetic inheritance, but still. And so there's this technology that puts it into a binary form, which is, I think kind of what you're saying. I mean, it's not binary, but it's quaternary, but it's digital. It's a digital compression. And then we have, for transmission between individuals, we've also had recently a revolution in information transfer by using a similar scheme of compression because it allows for error correction. And it just seems like the premise there is sort of this well, what I guess Dennis would call biological relativity, that there's no privileged causal level in biology, that causation moves between levels, and there's really no upper level. I'll stop right there because Dennis might have something to say, or Daniel.
Well, if I could come in just on the biological relativity, you would be amused to know that Richard Dawkins thinks that my use of it is tenuous and vacuous. I thought you'd enjoy that. Of course,
Richard
I have detailed answers because he doesn't understand the relationship between the various Einsteinian forms of relativity and the general principle of relativity, which has been around for a long time before Einstein. But that's another issue. You don't want to get into that.
So, because I kind of, I guess sort of look at the world as an engineer. I look at my one of my favorite engineers in history, Da Vinci, who said that all engineering problems are solved in the are solved in the animal body or in the human body, and just to extend that to how much we sort of instinctually have a sense of patterns of things that we then find or discover in in uh, um, in the equipment and the processes that we create. I'm thinking about how blockchain works and the way blockchain memory is distributed. And it seems to me, as soon as I learned about that, I felt like I've seen this before, I've heard this before, and it seems to me like we're at a place in sort of like human evolution where we can look at what's being discovered or emerged anywhere and then just say, do we actually already have that somewhere? And it seems like that process is likely what's going on inside the cell. I mean, we have so much membrane. We could have so much replication and duplication of information and a simultaneous updating of it. Think of all of the different capacities we have for sort of changing everything simultaneously. We have chemical and electromagnetic and harmonic acoustic. We have transfer of materials. It's really an incredible number of different methods. We have quantum, using the microtubules, or there's so many different ways that we could think about that.
Yeah, I'm comment on that, and then hearkening back to Wayne's very astute comment about exaptation. One of the principles that we know in biology, and I'm sure Dennis would back me up on this, is that nearly every thing that we can observe in multicellularity has its origin at the unicellular level. And
That's right.
that is actually how our system works. And actually, we have a form of proof that we rarely think about. If you agree that life began somewhere around 3.7 or 3.8 billion years ago, there was an extremely long period, 2 billion years, before you had eukaryota, your first instance of great creative novelty among cells. And even at that point, it took another 1.4 billion years before we came to what we generally agree was an explosion of multicellularity through the Cambrian explosion. Why? It's because all the hard work of evolution was being done in the unicellular state as it learned important natural learning skills through biofilms that could later become exapted to the principles necessary to support us as seamless holobions. And so the point of evolution has always been a continuous one, in which memory has been building over time, and we reach backwards and forwards across the toolbox according to our needs, and we redefine those consistently.
And that has to be right, doesn't it, Bill? Because we start as a single cell.
Absolutely. Yes. Thank you, Dennis.
It's got to have a hell of a lot there. Sure, some gets refined during development and interaction with the environment, but there has to be something in what you're saying. Absolutely agree.
Let me add one more thing. I have a peculiar point of view about biology, as Dennis knows pretty well. And one of the concepts that I've put forward with colleagues is called the 4 domains concept. What we purport is that, again, understanding the progression, the multi-billion year progression of life from unicellular to multicellular life, that the point of multicellularity is the unicell's way of exploring the environment, continuously exploring the environment, which is then returned through phenotype back to the zygote and redeployed within the zygote. But the point of all of this is the continuous support of the 4 domains: bacteria, archaea, eukaryota, and the virome. And the virome being its vital intercessory communications, a communication network among cells. And so if you look at things objectively, we're clearly not the point. The point are the 4 domains, because they are perpetual for billions of years. Species come and go over 5 to 10 million year intervals. They're transient in geological time, but the cell domains have been permanent. So they must be the object of the living principle, as far as we can understand it at this moment.
So we were both inspired by Albrecht Buehler, who said that the intelligence of the cell is both holographic or fractal and emergent property. Would you agree with that? The intelligence of the multicellular organism is fractal and emergent property of the intelligence of cells.
Absolutely. I would just say, I think the best way to think about this is the way William James went about it in the 19th century. And I know Mike Levin is particularly attracted to this definition. Intelligence is achieving the same goal through divergent means, roughly paraphrasing. It's that kind of flexibility, endless repurposing of old solutions to become new novelties and creativity that we apperceive as new form, but is largely supporting a single, continuous, coherent narrative.
So before Dennis leaves, I just want to say, Dennis, I would love to talk to you about getting the team of people that we have here. I'm working with Charlie and building Talking Octopus together to have a conversation about this new synthesis in a conference with published proceedings. I know, because I know Perry Marshall, that you've talked about this with Perry, but I think that it needs to happen. And we now are starting to get a group of people that know each other that I think would be willing to work on this. And I've put on conferences, so if you wouldn't mind, I would like to be able to reach out to you, Charlie and I, to talk with you about this. So sorry to interrupt, Bill. We should get back to your very important idea.
I'll just respond to that briefly, Mary. My problem is I've got negative time. We'll find a way. You can easily find my email on my contact page. So, or it probably is in what Charlie circulated. Try me out, but don't be surprised if it's going to take a bit of time to find a good slot.
Well,
Okay?
if you can find us a hoary old building with Gothic spires on the
Yes.
Oxford campus, and we could do the rest, that would probably be fine.
Okay, fine. Anyway, I must leave now. Thank you very much. Very exciting and very interesting and wide
Thank you,
range.
Dennis.
Thanks for joining us, Dennis.
Yes.
Oh, Wayne, you've had your hand up for quite a few.
Yeah. So one thing I think that we need to be careful about is that we're tossing around memory, the idea of memory, digital memory, and compression around a lot. I think it's really important to be aware. I mean, and this goes back to, well, William James, but more to Peirce and to Whitehead and to biosemiotics, is that there's a big difference between the way computers work with memory and the way organisms work with memory. For a computer or a digital system, a memory is objective. It's unambiguous. It means what it means. For an organism, memories are, sort of Mike Levin touches about this in some of the work we're going to be talking about next month. It depends upon context. It depends upon the interpreting system. A memory is something that has untold numbers of potential meanings, but what it actually means depends upon the interpreting system. Right? And so we have to be really careful when we use words like memory and compression, that we aren't getting ourselves locked into this, von Neumann architecture computational thinking on memory, and understand that when we're talking about biological memory, we're talking about this bow tie architecture and biosemiotics. Again, just kind of a general comment.
That's a great comment. I think it's really important to distinguish between memory and recording. And that's how I think of it, is that we don't really contain any recordings. Like our minds contain interpretations. And every time we return to the interpretation, it changes a little bit because we've changed a little bit. And our
DNA is the closest thing we have to a recording. That's where the
Right.
How do you guys know we don't have recordings? I think those membranes are just filled with mem codons.
Yeah, Mary disagrees.
That may be another.
I think we're our living liquid Polaroid is what I think.
The only possible issue, and it may not even be a real issue, is that with the DNA system and the genetic code and base pairing, you have a lot of fidelity to give you sort of these recordings or something that are effectively recordings over fairly long periods of time. I'm not sure whether you have that with membranes, or at least it isn't simple. It may be contextual. It might be that these, whatever you're calling them, these sort of, these little digital pieces of
mem codons.
Yeah, these mem codons. It may be that the entire environment which they contain sort of gives them a certain amount of fidelity. Although it's not clear to me how they would have fidelity over many generations.
Well, they could have a fidelity over many generations via vesicles. I mean, I was already thinking about that before I was introduced to Daniel and Dennis's lovely work, and the fact that there could be the way that we are able to reconstruct DNA is by overlapping the fragments, from DNA that's been fragmented. And then we just do a composite picture. Maybe memory works the same way.
By the
Maybe
way,
we have fragments of memories that are similar, and we have some capacity for sort of cohering the image by overlapping them.
Quick question. This is, I guess, for Daniel, who's doing a lot of work on vesicles. While we're on the subject, obviously the main cell membranes and some of the the interior membranes that are involved in organelles have various membrane-bound proteins and things like that in them. When you have these vesicles, are they always fairly pure phospholipid, or do they actually have membrane-bound proteins embedded in them typically?
Yeah, they're far from just pure lipid bilayers, so they're still just saturated with things, and it's a really important part of how they work. So the big kind of lock and key mechanism there are proteins called tetraspanins, and that's kind of how, in theory, and I suppose it's been demonstrated to some extent now, one tissue can choose the target tissue it's sending the message to, because it puts the correct tetraspanin protein on the surface.
So Daniel, do you know if anyone has done any work with looking at rafts that persist over time that might have something to do with memory? I know raft is the wrong term, because raft doesn't relate to memory. It relates to assemblies of proteins in the membrane for other purposes. But
so do you mean in the context of the brain and neurons, that kind of typical memory? Yeah. I don't know. That's kind of more specialized of an area. I'm sure that there's a lot of work on that. because a lot of what happens in memory is I'm particularly interested in RNAs, so I'm doing my reading, I'm focusing on RNAs. And
Yep.
RNAs get bundled up into kind of conglomerates of RNAs and proteins. And they need to be shuttled from one end of a neuron to another. And that often happens by being stuck onto lipid rafts. And then
This is exactly what I was imagining. Okay, I want to ask you more about this as I get more into the reading. Now, I've read your papers multiple times, so I'd at least be up to speed on those. Now, I wish I had read more of this stuff that I was finding in this regard. I'm very, very interested in that. That's very curious to me.
one of the favorite aspects of the paper that me and Dennis wrote was actually, maybe some of you remember or some of you forgotten, so I'll remind you. Sperm actually have lipid rafts on them.
That's right.
And what the purpose seems to be of these lipid rafts is to collect all of the vesicles coming from the environment, being sent from the body, and then to concentrate them in a docking bay underneath the sperm. And that's the site where they pull inside the concentrated extracellular vesicles on the outside, inside the cell. So the reason why I think that's so interesting is it shows that sperm aren't just kind of willy-nilly taking anything from their environment. They have been evolved and they've got adaptations to specifically, explicitly, by design, pull in information from their environment. That's what they're trying to do, right?
It sounds like they're collecting up all the family photos and they're putting them in an album to bring them to mom.
That's a really good analogy. It's exactly how I think of it. Yeah.
Okay, I feel so vindicated.
So can I ask, can I zoom out a little bit, Daniel, and ask you a question? Because I think there's an assumption that maybe separates me from most people who are interested in this, which is maybe because I'm just too simple-minded. But okay, so what we have here is an example of the phenotype level, the individual cognitive self instructing the single cell, right? And instructing the heritable material. And so at the level of the heritable material, we have an explanation for why it's coherent, natural selection, right? I mean, it's gone through, this process that weeded out things that weren't coherent. But if instructions are coming from the phenotype level down to the genotype, to the heritable material, then there has to be another mechanism that's making those instructions coherent. And my assumption here, I think I've isolated my assumption. My assumption is that coherent function requires coherent structure. And some people seem not to have that assumption.
Well, yeah, I don't either. But here's the thing, so once you have these vesicles involved, right? Now you have all kinds of potential, especially with the sperm. Now you have all kinds of possible imprinting, silencing, or activation, things that affect chromatin structure that can be transferred directly to the germline. So even if they don't change the coding sequence, they change the expression. So in terms of what happens in development and what happens later in life, this is heritable information.
So why are they coherent? Why are they coherent?
Because the whole thing is a coherent system. I view the genome as a little toolbox that's part of a coherent system, which is at least the entire organism. Certainly, yeah.
I agree, but why is it coherent?
The answer, if I may offer, is you have to be coherent in thinking through the cognitive frame. If life begins with cognition, which is what my colleagues and I assert, then natural selection is not how evolution proceeds. It proceeds through cognitive selection. If life is problem solving through communication, like Arnold Duluth would say, or Popper, or any number of people have asserted, then the issue is to be coherent with your initiating assumptions. And so cognitive life solves problems, which means survival, reproduction, and more than anything else, maintaining states of preference. That's what it wants to do. That's what life is. And it does it through selecting its tools, and that's cognitive selection.
But Bill, survival and reproduction is not in the cognitive domain of an individual.
Preferences.
I can't learn from survival and reproduction because I only do it one time. So it's not in my cognitive frame. Like, it can't be. because my mind has never experienced.
No, you're right, but put it in the cognitive frame. You are choosing your tools to sustain states of preference, which is homouretic flux. That's the dynamic flux that every cell wants to maintain, which correlates with the state that it will be found in most commonly if you examined it at a snapshot. The issue really clarifies when you realize that cells are seeking to maintain that preferential state. It then it selects its tools to sustain that over different kinds of environmental stresses. And the environment, natural selection exists then as we now can correctly see, as a post-factor filter of the selection of those tools. So the organism is sustaining, its mission is to sustain its state of preference. And in so doing, the environment requires it to be survivable and capable of reproduction. So it's learning through progression, through surviving the environmental stresses. In effect, the environment tells the cell that its measurements of the impact on the cell, its measurements that it has made to support its preferences are correct over time. And a cell learns it can better support those preferences collectively than individually because all information is ambiguous. They never have any, the comment that Wayne made that computers and living systems are entirely different on the basis of memory is absolutely correct. It's a terrific insight. But the reason is that the forms of information that they have available to them are entirely different. Digital systems are square waves, and the simplest thinking process, square waves, and living systems are analogs. There's ambiguity on every single thing a cell can know. There's no such thing as a pure light source. It has to transmit through media. It has to go through a complex discriminating membrane in order to be measured inside the cell. So a cell can never know an absoluteness about the outside. It's always ambiguous. That's why we have multicellularity. It's the wisdom of crowds. It's how we solve our own problems, because we are always fractal reiterations of initiating unicellular pathways and patterns that have yielded enough success to continue the trajectory.
But I think cognitive problems are different from problems of survival.
No, they're not. They're one and the same.
I don't think so. I mean, a contradiction between 2 points of view is a cognitive problem. It doesn't have much to do with survival. And I think organisms also take on lots of projects that don't increase their survival, obviously.
Yes, because you and I are in agreement. Because the organism's major role is to try to continue to exist within states of preference. Just examine your own life. You're continuously reasserting your states of preference in everything you do, including being on this Zoom. I really believe it's illuminating to think more about preferences and less about survival and reproduction.
Wayne?
Yes, Charlie. So, I think in a way, you guys are both right. And here's the tie-in. So the single cells all have these preferential states they want to maintain. But this scales up over many different scales. So depending on what scale you look at, you're going to see different preferences. And we also have shown or have seen here that there are ways that there's interaction between these scales that feed up and down. So the preferences at a higher level, once you have, say, a human being immersed in a cultural and symbolic environment, these preferences are very different than just survival and reproduction. However, there's feedback across levels, there's feed up and down, this whole Neoplatonic view of reality. And there's actual mechanisms we've been showing here where, using epigenetics and things where these things feed back over many different levels. So it really probably is preference at every scale. And there's ways that those preferences can go all the way down from the higher level organism all the way down to the genome.
Yeah, if you
But
allow,
it kind of has to be like, I'm sorry, go ahead, Bill.
yeah, just the issue of preference versus survival and reproduction. I think it's a very, very interesting question, and I think you're absolutely right to ask the question. And I will answer that. We've just run the first experiment of its kind unintentionally. Clever humans are the first opportunity in the history of organisms to disconnect reproduction, the act of sexual, disconnect sex, the act of sex, from active reproduction. We're the first, actually, couple of generations in the history of the world, of this planet, to be able to do that. And what do we know now? There's a baby bust in many European countries. People are choosing, men and women are choosing to have just as much sex as they had before, but they have been able now to voluntarily disconnect it between reproduction. We've basically known this anyway, because 15-year-olds don't have sex together in order to produce children. They have sex to have sex. And so preference comes first, and reproduction is its issue.
But the preference is not just preference. Like, it's not agnostic. Like, it's structured preference. Like, it's a cognitive, like, I'd call it explanation, right? And that's why I think it's a totally different, like you said, natural selection is post facto. So it means that natural selection never determines the aims, the preferences. Like, they're determined by something else. And here we have an example in this paper of a whole, like, very involved evolutionary apparatus for, like, literally sending these messages and then capturing them in the germ cell, which is just, like, totally wild. I mean, it's as if the whole system evolved for the purpose of communicating this marginal changes, these tiny marginal changes in the phenotype from one generation to the next, which means that has to be the source. The source that's the configuration that's sending the message has to be coherent in the first place. It can't be downstream of another process that, if it's sending the marginal change, it's the source of coherence. And so if that source of coherence, only went through natural selection post facto, it's got its own endogenous source of coherence, which I think is cognition, like explanation. Like certain things, like when we have a mind, even if it's only a single cell, it has a mind, which forms an explanation. And then there's certain observations that don't fit that explanation. And those are problems to be worked out that then, it reorganizes itself.
Yeah, I think
like reorganization has to happen for a good cognitive reason, like within the cognitive domain.
Charlie, I think you're really hitting it on the head. There's a vital separation that we're required to think through each for ourselves. My colleagues and I state very directly that life began with cognition. Life began with that, and we don't know how or why, but with the ability to know uncertainty and to want to continue. And then I've added in a preferential state. That's the source. It always is coherent back to that source. You have to decide whether or not you will accept that life and cognition are coterminous. But once you do, then being coherent is carrying that through in every single step, in large and small things. So there's no natural selection per se. There's cognitive selection with the environment as a filter. So biology changes absolutely dramatically if you're willing to exceed one initiating principle. Life and cognition began together. And if you reject that, then we can then you're talking about the emergence of cognition, and that becomes a whole new set of questions. But once you start with that unity, then I'm saying that there is a very coherent way of understanding biology from that moment forward.
Just very quickly, because I'm going to have to go. My wife is telling me we have some stuff that we have to do. You know, if you take the white, just very quickly, I don't know how familiar you guys are with Alfred North Whitehead. I know Mary is, but not everyone is. If you take the Whiteheadian point of view, this really makes very deep sense. And actually, Whitehead saw deep continuity from the entire universe. Maybe not with cognition the way it's like, so organisms, what he would call societies of actual occasions, ramped up this sort of, I guess the word is, you know, panpsychist is not really a very good word, more pan-experiential. You know, so for Whitehead, it was really all about experience and aesthetics, which has to do with preferences. And this goes all the way down. But with organisms, you have just a ramping up of this effect to a really high level. So I just want to throw that out there for folks that are familiar with Whitehead. I think you know you're on the right track when you say it's all about preferences, or whether you want to call them preferential states. But it may very well be something that goes all the way up and down.
Wayne, you know that I agree with you on this point. I would love to talk about this more, but I have some questions for Daniel while we have Daniel's time.
Yep, I actually really have to go, but unfortunately,
Okay.
But, love to do this. This was a really great discussion today.
Good to
Daniel,
see you again,
I
Wayne.
have a question for you.
Thanks, Wayne.
About which vesicles are sort of chosen. So where's the sorting? Where's the sorting hat? And is there anything, are we capable of recognizing any kind of sort of like quorum or you know, method for, or is it just entirely stochastic for which vesicles get chosen by the sperm? How do we know what's going on there?
It's a good question. I think part of the answer was already alluded to before by talking about, is the membrane of the extracellular vesicles just a pure lipid membrane, or is it also saturated with other molecules? And we know it's filled with other molecules. So it's something to do with those molecules. And if there is a fit between the lipid rafts on the sperm and the proteins they have there, and the proteins on the extracellular vesicles. It's actually funny you bring this up, because one of the reviewer comments we got for the paper was, how do the sperm select which EVs come inside?
Right.
And actually nobody knows, except for just talking about generally how any tissue selects which EVs come inside. And that's to do with this kind of lock and key.
It has to do with the membrane, gating.
The complementarity between the proteins there. But I think it's actually really crucial, because if it can be shown that some tissues, let's just say the brain for one example, packages EVs with proteins, which essentially say these EVs are going to the sperm specifically.
That's right.
That's really showing the evolution of a brand new inheritance system.
So is there any way to isolate the EVs and just see what, even in the crudest way, what properties they might have in common in terms of size, in terms of the concentration of certain proteins, even in the number of like aromatic, residues in the proteins? Is there anything?
I mean, this has been done a lot. I suppose it's harder than one would imagine to work out the complementarity between the origin and the receptor tissue, because there's not been any work done as far as I found. I couldn't find any when I was trying to answer this reviewer's comment about how specifically how the sperm do it. And I wonder why, if it's because it's, in biology, it's never clean cut. It won't just be that there is one protein for the sperm. It will always be that there's 50 or 100, and there's a kind of a complement of these things, which each tissue and maybe even cell types within the tissue are sharing. So there'll be
Right.
a clustering of these things. So we need lots more data to be able to deconvolve those.
I'm just thinking of, it just seems like there's processes that are replicated, across species, everything that's got cells, there seems to be this like resilient pool of possible means of communication that arise and then go back into sort of like just memories, you know, for future reference. I'm just thinking of the way that bees, communicate with one another. The bee that comes back and does the waggle dance, that's the longest and the most energetic, is communicating, that, no, I really found the flowers with the good pollen. And there's a lot of them and they're close by. And I'm just wondering if there's something to do with, a protein that shows up more frequently when information is more important. You know what I'm saying?
Not exactly.
Well, like if there's a protein in the raft, that there's more of that protein if the brain thinks that this is really important information to be. Do you see what I'm saying?
I see what
The
you're
form
saying.
sensing idea that there's more of something to indicate importance, or there's more energy, or the cell has the vesicle. I mean, I don't know if the vesicle, well, structured water is able to capture and release and send on start the oxidation reduction process just right in the structured water. If there's something, there's more energy, there's more of a protein, there's
Yeah,
there'll be,
I think I understand better what you're talking
yeah,
about now. And maybe it's to answer the question in a different way. So you initially answered the question, how are they packaged? How does the
yeah,
selectivity come around?
yeah,
So one way that can happen is at the receiving end of the sperm saying, you
yeah,
can come in, you can't come in.
right,
The other way it can happen is at the sender end. And the sender can say, we're sending this parcel. Just by default, we always send parcels and they always go to these recipients, but something's kicking off, something's happening right now. This parcel, this time we're flooding it with information. That
yeah,
might be the waggle dance thing. The
right,
parcel was always going to be sent, but this time we're cramming it with information, which essentially says, waggle, 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 here's your information.
right. And that could be done by giving it more, more of a potential oxidative energy in the structured water, or it could be that there's lots of vesicles of this type. And I still think there's something to the idea that each vesicle may be will obviously be slightly different, but they create a sort of an overlap, which gives a holographic image, that the sperm would think was important.
I see what you're saying. For me, I just care about most of all the RNAs. So if there was, to stick with the
Right.
holographic image thing, it is possible that when an RNA, particularly a non-coding RNA, which has been transcribed in response to some stressor, let's say diet,
Yeah.
and that transcription of that non-coding RNA is actually informing the rest of the body, not the germline,
Yeah.
how to respond. In a sense, that is a holographic image of how the
That's
body
right.
should look in its response.
Right.
And it just so happens that it's also going to the germline, so it's being passed both horizontally and,
Right.
and vertically. But for me, I tend to think about the contents of the EVs in molecular form. Which nucleotides,
Yeah.
how are they folded, are they modified, and these types of things.
So, do you ever see a situation where the RNAs are not exact sequences, but they're overlapping in some way? Sort of like when we're sequencing nucleic acid sequences with you know what I mean?
Yeah, I know what you mean. I would say yes, probably. So if
Yeah.
you're looking at which RNAs tend to be found in the EVs going
Yeah.
to sperm, there's a real enrichment, over a representation of fragments of RNA.
That's what I'm saying. And the fragments are, if you lay them, it's like how you sequence DNA is you lay the fragments. Yeah.
Yeah,
And
so
it's
it's actually a really important aspect of what they're
Yeah.
supposed to do when they get to the other side. And
Yes.
it's this overlapping thing. So the kind of how the theory seems to be going these days is you have these fragments
Yeah.
coming from the sending cell,
Yeah.
then they go to the sperm, and then they overlap their original
That's right.
transcript, and they either turn it off, they get in the way, or they connect it to some other RNA, or they connect it to the DNA. So this overlapping aspect is a big feature of how they connect RNAs to other RNAs, how they connect RNAs to the
Yeah.
DNA, or how they connect RNAs to the epigenetic machinery of the cell.
So this leads, so since I think of myself as a natural philosopher, first and foremost, but a lot of what I observe is that we don't really understand anything unless our cells are already doing it. And that it's a matter of recognition, which goes back to, I guess I'm a Neoplatonic in that regard, that it's all about remembering. And it just seems to me like there's so much capacity going on in the cells that we just know because it's literally in our bones, it's literally in our cells. And that this whole process of using this process of overlapping fragments to sequence DNA in the first place is because we already know it so well. So sorry about the soapbox, but that's my own personal philosophy as a natural philosopher.
Well, can I just say, like, to agree with you, Mary, to me, this paper and its implications are that is the sort of the starting point is the mind. Like when we take a psychological explanation of life, like we ask what are we doing? Why are we doing it? We say we're doing it because we either love to do it or we fear something else. We're afraid of something else. So it's emotional. There's an emotional explanation for behavior, at least on the the level of the phenotypes we're most comfortable talking about, which are adult humans, and everything else is an alien as far as we're concerned, including small children. But there's this emotional starting point that then becomes physical later. And like, here's the mechanism. Like, this is literally the inheritance. Like these rats, like I thought about your papers because Kate Kaufman, who hasn't said anything, has a beautiful paper about the biology of emotions. Because it's literally a direct inheritance of a fear. Like there's an acquired fear of a certain chemical that then it even was inherited when they made a test tube baby with the male rat sperm, and the babies inherited this fear directly from an acquired experience that the father had. And so it means that the emotional, it's like Richard says, it's all about love. Like the organism, the physiology and inheritance of the organism is acquiring all of its teleology, all of its rationale, all of its what it wants to do. It's acquiring all that from the emotional experience of the ancestors directly in a physical way, like through a known mechanism.
And to tie back to this idea of valencing, the importance of the information that's coming through, Charlie, you and I have talked about the fact that even that the chiralization, the creating of rotation of the light in as it moves through the cell happens in lots of different ways. And it seems to me in this picture that I'm getting of the membranes as having these multiple layers, a three-dimensional layer of capture of and transduction of information. It feels to me like when we say you put a different spin or a different slant on something, it almost feels like, again, the cells are telling us that it valences information by changing the polarization, that's the slant, or the spin, the rotation of the light as it's moving through the membrane. And because the membranes are active materials, they can change the shape of proteins or bring up proteins that will change the chiralization of the light, and then that can be captured in some way. I know that this is a very arcane theory that I'm working on, but bear with me. It seems like there's something in there about the colorization of emotion by changing the chiralization in a change, using chiaralization to change the rotation of light.
Well,
Can
I mean,
can
I
I
know,
respond to that?
Catherine, does it sound crazy to you?
Oh, it sounds perfectly wonderfully crazy. Can somebody tell me how to raise my hand? I don't know how to do that. But firstly, I want to say, I think everything that you've communicated, and you clearly thought about it very deeply, Mary, it fits with what I see happening with emotion as something much, much deeper than neuroscientists would like to believe. And when you talk about polarization of light, I mean, you want to take this all the way down to, well, emotion is centered upon what is self-relevant. It's all about a self-reflective act that's very, very, very deep, and how identity is made distinct. And that's a huge question there. But when it comes to the feeling and the behavior, those things are coupled together in our emotional system. They're not separate systems, even though motivation is usually like, they're not separate. And you can see this machinery on the cell membrane of the simplest organisms, meaning that there was some information, semantic, evaluative information that was built into our learning system, into our everything at a very, very, very bottom.
Basic level.
Yep. So I argue that the core of sentience, and perhaps even qualia itself as a category of experience, setting consciousness aside, was the very first distinction was something that felt good or something that felt bad. And that can go all the way back to preference, optimal energy, least action, edge of chaos, all of that stuff is part of this beautiful binary symbol, because all training systems, all learning systems are based on that, what I call the fundamental semantic information bit, which is the distinction between a feel of good or a feel of bad, a feel of pleasure, a feel of pain, two things that are contrasting, because then you set up evaluative categories, and then you have Pavlovian learning. And learning is central to memory. I mean, we got to really look at the learning process before memory, or we want to define what kind of pattern making processes endure, and why they endure, and how they change, and how they transduce in the whole flow of reality, if you will. But that I really like what you're saying, because if you want to try to think about agency and factor it in all the way back. Stu and I are working with a quantum physicist right now on a reconstruction, of reinterpretation of quantum mechanics that puts agency, sort of an agent back in the machine. And when you follow it all the way down, you think about energy and work, and the relationship between, energy is the capacity to do work, and work is a release of energy with constraints. That's very circular. But there's actually an agent hidden in there who's doing the work, where are the degrees of freedom that are being, okay. So if you follow that all the way back down to like Conway-Cochin free will theorem and stuff like that, Cochin has written an update where he basically talks about the rotational degrees of freedom.
Okay, so the exact so the way that I see it is that the way that I'm seeing it is that everything is either dextro or level, right? All the proteins are dextro or level, and water actually is, even structured water is actually chiral as well. And what if we actually have a binary there? And what if we have fear, which is going to take the normal left-handed rotation and turn it to the right? And we have love, and would really have a basic binary of fear and love, and then we have something else which is adding the other qualifications of that. I mean, I'm thinking all the way back to Candace Pert, with the neuropeptides, we've got them floating around everywhere. What if there's something like that? Or what if it's the RNA that would code for those peptides, which is also creating this quality, this qualification? We should talk more about this, Catherine, because I've been a lot.
I want to throw out there that thinking about this a lot, and Candace was an early mentor as well for me, and she.
Oh, wonderful!
I think the concept of resonance and dissonance is getting closer to what you want. Phase relationships, and constructive and destructive interference is what we're really talking about. Because
Yeah.
if we have the ability to shift our rotational or, change our spin, it's not just spin up or spin down, it's all kinds of different things in between that might very well have much more meaning than before it spin up.
Well,
or
that has to do with the structured water and the shape of the the peptides and the oligosaccharides.
Yep. What she calls
And
the
three-dimensional
peptides,
yeah.
peptides are molecules of emotion. And, peptides are just really very, very, very short proteins. And they were some of the very, very first molecules that our receptors pick up.
That's right. There's some of the early ones.
But
And
you're totally onto something with light. You're totally
Yeah.
onto something with light.
I think
And
so.
talking
Well, the
about
fact that
earlier-
the different
Just one more thing
peptides.
out. One more thing out. I've always been curious about the double slit experiment, okay?
Yeah.
The detectors do this, the mirrors do that. And when it really comes down to is that there's two things that can happen. Light can be deflected or light
Yeah.
can be transduced. And
That's
those
right.
are the 2 degrees of freedom that we really need to be thinking about in terms of how does that work? And it sounds like you've done a lot of thinking about it. Anyway, that's enough for me. But do tell me how to raise my hand.
You go down under react where the heart is. Catherine, I wanted to say that I would like to go back and look at the molecules of emotion and analyze them in terms of the chirality of the amino acid prevalence in them and whether or not they're aliphatic or aromatic, because we have different rotational effects happening in those. And I think that this is something I've been learning about from Dennis Kent, who's been using light to turn on and off molecules. I wish he'd been in this meeting, but there's something about the difference in them that is literally the color that we're getting from the residues.
You're right about the aromatics. In fact, I have a friend who studied the whole archaea and everything. And so
Yeah.
the aromatics were one of the very first kind of
That's right.
things. But I
And they are, and
argue
they're
that
also,
the valence came before all of those other complex systems, although optics may very well be part of
it feels like it starts with optics for me when you consider the fact that if water is actually if structured water can act as a negative for the creation of other molecules, and water has the 5 and 6 ring structures in it in the structured water, then that would make it more conducive to be creating a negative for. So anyway, let's talk about this more, Catherine.
Thanks.
I think this may be a good place to trail off, but before we do, I just wanted to say, Daniel, this paper has just opened up so many fascinating avenues. I'm
so grateful for this work.
We've been kind of speculating about this, and it connects to like Dennis's whole philosophy about harnessing stochasticity so well. We've been trying to, like, in this group, continually speculating about like the sources of stochasticity and the sources of harnessing. And this is like, for me, it's just like, not that we need it anymore, but it's like really good empirical proof that, the phenotype is in control, that the organism is in control of its inheritance. And so, this is just really, really super welcome. And thanks for doing all the hard work, to put this out there.
Yeah, thank you. I had a really good time talking with you all. I've come away thinking about the paper again. I've not thought about it for a while, in lots of new and interesting angles. And I think I have a lot more work now to do after this discussion. Yeah,
Yeah, I mean, it's really like infinite amount of work you're going to have to do to totally re-explain biology on some different basis. So next month we're having Mike Levin, and we're gonna go into like the nature of memory, and I think this is a really good way to lead into that because we're all like, I'm now thinking memory is basically everything. And like there has to be a stochasticity and a harnessing in every biomolecule, the reason why it's in the place it's in and the reason it's doing what it's doing all ties back to memories of prior organisms. And so, what created those memories, like what process filtered out the things we don't remember from the things we do remember. Like that's the cognitive glue, the cognitive filter, like Bill's calling it preferences, which is, I dig that. I mean, I think that's probably a more like palatable framing than the one I have, because I tend to be like really what's it called? Not anti-clerical.
Radical.
Oppositional. Yeah, radical. Yeah.
Yeah, I agree.
Totally.
I think Mike Levin, to follow this meeting, is a very good contrast, because learning and memory, particularly memory, has been a foundational concept in this discussion. But it's been based on the idea if you've got DNA and RNA, they're both nucleic acids at the end of the day. Whereas all of Mike Levin's work is basically that all learning and memory and plasticity is above and beyond the level of DNA and RNA. So it's both of these types of things lend themselves to memory and learning vocabulary, but in totally different ways. So it'll be an interesting to slide right into that discussion.
Totally. And he also is kind of way more bottom up than you guys, I think. He's like entertaining the idea that memes are sort of, causal in themselves, like thoughts are thinkers. Like there's this idea that thoughts themselves are driving the bus, which I disagree with, but I think it's a really interesting like contrast.
Charlie, I see it as the difference between energy and the imprint. So if you think of energy as information, and then there's capturing of that in an imprint, which is then reactivated and then becomes information again.
Right.
Like if you think about it as literally the reading of my holographic Polaroids, right?
Yeah.
Then it becomes information again, and that is transmitted as energy.
Yeah, I mean, it definitely is like coded and decoded and then coded again over and over.
Daniel, I'm so grateful that you presented today. This was exactly where my thinking was at, and I had all of these weird questions to ask somebody.
Well, you had a lot of questions, which was great for me. So,
and after I talked to Catherine, we're going to have a ton more.
I had to revise reading that paper. I've not read it in a while, so I actually don't work on this area anymore. I've moved away from this area, and it was kind of a shame. I wanted to do my PhD in this area, but it just kind of didn't happen. I ended up time goes on, and I drifted away to something else. So it's nice to return to this, which was my first love, really, in biology.
Well, then go back to it. You're young. You can still do it.
I can. Yeah, I don't know. I have other interests.
Get another postdoc.
Yeah, well, I probably will do something like that, but it's one of the main ideas or interests in my constellation of passions. So it's nice to come back and have a really, really engaging and full-on discussion with all of you. So thank you.
Come on, just put together a research proposal. Charlie and I know all kinds of tech billionaires now. I'm not kidding at all.
Deal.
Rafe, you can weigh in now.
He's already gone. He's
Well, I know Dennis is very keen. There's certainly enough ideas and plenty that need to still be tested. I think it's just finding the time and finding the resources and the people who have the willingness to try and do them. So,
yes.
I'm discovering in my reading and just reaching out to people that the people who wanted to do all of this stuff are now material scientists. And you scratch them a little bit and they're all like, oh, I would love to actually work on this.
There was somebody, it's a shame he didn't come today, Edward Wills, I think is his name. Is that his second name, Charlie?
Edward, right.
Yeah. He was also here in Oxford, knew Dennis as well, but he was actually working hands-on on EVs, doing this type of research, and he was going to try and come today, but I suppose he couldn't make it, which was a shame. But it means that in the future, when we discuss again, and if he is here, we have even more things to discuss.
That's really great.
Okay.
Thank you, guys.
Thank you all. Nice to see you all again. Bye-bye.
See you later. Bye-bye.
Bye-bye.